Okay, and welcome to step 10 of glycolysis. So what would happen here is that we have a very high energy intermediate. Want me to remind you why this was high energy? Yeah, this is a phosphate group attached to COO. This one looks more or less like a phosphor ester, linkage. Look more or less like that. So when you break this one, a lot of energy can be released. That's why this one, PEP, is called a high energy intermediate. Right, so PEP is going to be converted to pyruvate. And what happens here is that the phosphate group will be removed. So guess what enzyme is this? Of course, that will be a kinase. A kinase enzyme that acts on the pyruvate. Well, sometimes they name the enzyme according to the product that will be formed as well, not just the substrate, okay? So this is pyruvate kinase enzyme that will be used, the kinase that will create the pyruvate, PK. There you go. That's what the name means. And they remove the phosphate into what? What will happen to the phosphate? There will be ADP in the environment to accept it. It will always be like use up battery, ready to be recharged again. So they accept the phosphate to become ATP. So this is the second ATP generation. So that's not the end for it. There would be some H+ in the environment that will attack into this double bond. Double bond is always a target for a lot of chemical reactions because obviously they are polar and there are 4 electrons there. They attract stuff to attack in there. So when H attack in there, it's gonna go. The only place is to go to this carbon because this carbon is full already. Okay, so it attack into here, so becomes CH3, and then that extra electron jump to there and then create the double bond. Alright, so this one jump into there and then causing the shift of electrons to here and then finally to there. This phosphate, when it's removed, this oxygen already has 1 electrons freely, so it will move 1 electrons into here. So you see the movements will create a double bond. Okay, so this one here and then electron freely move into here and then become double bond. Okay, this is a pyruvate. Exactly the same thing that I told you in the last episode. Okay, so from 1 glucose molecule you can create 2 pyruvates and then 2 ATPs. Okay, from step number 6 I told you that the gap, the GAP, one gap will become 1 pyruvate and then 2 ATP, right? That's what happened from step 6, 7, 8, and 9, 10. 2 ATP from 1 gap. But then 1 glucose, you have 2 gap, 2 GAP. So it's supposed to make 4 ATP by the end. However, in step 1 to step 5, we already use up 2 ATP. So by the end, the net is that we create 2 ATP, okay? Okay, if my explanation is a bit too fast, please, go back the video and then you will see it. I try to make the explanation as easy as possible for you to understand. And I hope the whole series was helpful for you. I hope you share and like this video, and please subscribe and then click on the bell button to get notification about new videos, okay? I'll see you. Bye bye.